What's up, penguins? Today we are going to do a Unit 7 Q&A on natural selection. So as we've done the previous couple ones, what we'll do is we'll do a quick review over the information. And then after that, I'll go through the questions that were pre-submitted. If you have questions along the way, you're welcome to throw them into the chat. I'm going to answer them as I go. But again, we're focusing on Unit 7. So if you ask questions that aren't on Unit 7 or aren't just on overall, just like test information, then I'm probably going to have to just, wait until later a session for those. So as a reminder, I've got a ton of free resources for you, all on apbiopenguins.weebly.com. I currently am doing Monday through Friday, review on my Instagram. And so when you go there, I'll have questions in my stories. I'm currently almost done with Unit 7, so there's really a small amount left. But the good, valuable thing that's still available is that I have a question box at the end. You can put your questions in there, and then I'll answer those questions after I do the review each day, which is kind of helpful for some students to get those questions answered. On that website, you're also going to find there's a 396 page review guide that's got topic questions, but what you probably want right at this point is that there are all of the multiple choice questions and all the free response questions are organized by unit. So if you're saying, okay, I need to practice Unit 7 multiple choice questions, you can go to that review guide and find Unit 7, and you can have some released multiple choice questions from College Board, as well as you can have all the free responses organized for you, which is really kind of helpful. There's also 120 review games on there. There's FRQ Fridays where I go over every single FRQ from 2013 to current, excluding 2020 because those weren't released. And then there's tons of other stuff on there. So, just go click around. You'll find a ton of stuff for you. And again, everything on the website is free for you. So we are, as of tomorrow, it will be 3 weeks until the AP exam. Yes, 3 weeks. So we are past the 4-week plan, but I do have a way that we can modify the 4-week plan. So if you were to start today reviewing, that gives us 22 days for the exam. So you could use the 4-week calendar, but you're going to have to shift the day slightly. So all these breaks will go away. You will not get these break days anymore. And then you're probably going to need to combine Unit 1 with the CED quizzes with their Unit 1 review content. And then same thing with Unit 2. That would catch up with 2 days that we might be missing. That would kind of help us out to get some of that catch up that we might need to do. And again, the AP exam is on May 5th. So in case you don't know when the AP exam is, it is on May 5th. So today we're going to do a little bit on natural selection. We'll talk about Hardy-Weinberg. We'll do phylogeny and kind of evidence of it. And then there's some Unit 7 Q&A that was already pre-submitted. But again, as you come through this, you'll find that, I have a question, put in the chat. I'm happy to answer it as we go. So selection, natural selection, what is the theory? So this theory came from Charles Darwin, and its whole big thing is that those individuals that are more favorable are going to be more likely to survive and more likely to leave more offspring. That's the big thing you're going to have to always say in these responses whenever you do a free response on it, is that you're saying that some trait was more favorable, and because that trait was more favorable, they were more likely to survive, and then because they're more likely to survive, they left more offspring. So it changes the allele frequency, and it kind of makes it so that there's more of those alleles in the population. I also recommend not studying the day before the AP exam because of the fact that your brain needs kind of a day to kind of refresh. So if you were noticing that on the calendar, I had a break here originally, I still, think that you should probably take a break on that day. Okay, so some big evidence that we have of this is peppered moth. Okay, so the peppered moth experiment, actually it wasn't an experiment, it's really what happened. Before the Industrial Revolution, there were these moths, and the moths were kind of a lighter shade of color, and they blended in with their the trees. Okay, and so due to the Industrial Revolution, the trees kind of got a little darker, and because they got darker, those moths that were lighter were less favorable. They were more likely to be eaten, and so we saw the allele frequency of those lighter moths decreasing, and we saw the allele frequency of those that had a darker color increased. Okay, so it's not that the Industrial Revolution caused the change in their frequencies or the change in their colors. It was a mutation, and because of that mutation, that allowed the darker ones to be more favorable, so they're more likely to blend in and less likely to be consumed. So again, more likely to survive, more likely to leave more offspring, so we would see an increase in that allele frequency. Same thing with antibiotic resistance. That's also looking at natural selection. Some students get confused and think that's artificial selection because, we're introducing those antibiotics, but again, it is how they're changing in the environment. And so With antibiotic resistant, there is a bacteria that has a mutation, and because of that mutation, it's going to be able to be resistant to the antibiotic, this drug that's designed to kill the bacteria. And so, because of that resistance, that bacteria is going to be more likely to survive. And so, we'll see an increase in that allele frequency. So, someone says, what's an allele frequency? That's just looking at the number of the alleles over the total alleles in the population. So, if we say that there's 75 dominant alleles over 100 total alleles in the population, the allele frequency would be 75% or 0.75. So, we're just seeing like what is the proportion or the frequency or the how many of them really there are in the population. So here again, we're looking at natural selection. So we've got the little beetles, though, that are more likely to kind of blend into the rock background, more likely to survive. And so we'll see an increase in that frequency. So here you see the frequency of A was 0.3 originally, and here is now 0.6. So that's what I mean when I say that we have a change in that allele frequency. And so one thing to be careful for is Lamarckian statements. So sometimes students kind of fall into these Lamarckian statements. So in case you don't know, Lamarck was a scientist, and his big push was that if you had these giraffes, and the giraffes stretched their necks, and because they stretched their necks got longer. And so each generation of giraffe is going to have a longer neck. Well, that's not true. We know that the giraffes that had longer necks due to a mutation were more likely to survive. And so we saw that over time, giraffes got longer necks. But his big thing was that because they stretched their necks, they got longer. So be very careful of talking about inheritance of acquired characteristics. So in terms of that, just because you gain some trait during your lifetime doesn't mean it's being passed on to your offspring. It has to be in those gametes in order for your offspring to get those. Antibiotic resistance was added to a plasmid is that artificial selection. That's more of a biotechnology and is more of genetically modified. But again, it's going to still be kind of, I wouldn't really call it natural selection, but once it's introduced into that bacteria, it is natural selection that's allowing it to increase. So I still think that we're looking at genetically modified organisms, but still nature selecting for the bacteria that is more favorable. And we'll see that the population will increase in terms of the number that are antibiotic resistant. So we didn't change every single one of those bacteria. We only changed that one or that, that small colony, and then they created the whole bigger group. So I'm going to still go natural. So artificial selection, this has to do with the organism certain traits are bred until that population has that trait. So if I want a cat that has curled ears, I'm going to keep breeding these cats until I finally get two cats that have curled ears, and I'm going to keep mating the curled eared cat forever and ever and ever together so that there are no other trait other than that curled cat. So for example, we have dog breeds that are bred for certain traits. The corn for maize, so originally maize used to look completely different, and now it's been changed so that it's kind of the more kernel shape that we have now. Wild mustard, we took different components of the wild mustard, and then we made different parts. So like the leaf became the kale, cauliflower was, I believe, the apical buds. Like I can't remember all the components, but we took certain parts and then made them into the parts that we now eat. So here you can kind of see that originally we had the wolf, and over this artificial selection, selecting for certain traits in them, like, the ability to look for these small rodents. Those could be like our fox hunters. And so we kind of selected certain traits and bred those traits out. So in terms of our selection, we also see that we have three ways that things can be selected. Okay. So disruptive selection has to do with that the two extremes are being selected. So if this was our original population, we kind of had, the intermediate was the majority. There's some type of disruption, and due to that disruption, we see an increase in our two extremes. That would just be the two phenotypes that are found on the outside. So if there were three different types, beaks, a large beak, a medium beak, and a small beak, okay? And there was a drought, and the drought, I can't, like, it's going back and forth between the two screens to move it, causes a lot of issues, so I can't move my face. So we have the large beak, the medium beak, and small beak. And so if there was a drought, and so now we don't have this medium seed, okay? So these small beaked birds are going to be able to get the berries and the smaller things. And the big beaked birds are going to be able to crack these hard, large seeds, okay? That one that has the medium beak is not going to be able to compete with these small beaked birds to get the berries. And it's not going to be able to crack open the seeds that the large beaked birds can do. So we're going to see a decrease in that medium beaked kind of bird, okay? Stabilizing selection, this has to do with if the intermediate is more favorable. Okay, so there's something about this intermediate that makes it more favorable. The biggest example I can, or like the one that always comes up to mind is childbirth and child weights. So if a baby's too small, like again, we're thinking evolutionarily, right? So if a baby was born too early, then we're going to see the baby's probably not going to survive. And if the baby kind of is too big when it's born, then it's probably, also not going to survive. So we kind of see that most babies were born in that 6 to 8 pound radius. And if you really question it and you kind of went full term 40 weeks, ask your parents, what was your birth weight? And I guarantee you were probably somewhere close that 6 to 8 ballpark of life, 6 to 8 pounds. I know that I was like 6'15", I think, and my kids were somewhere in the sevens. So directional selection, this has to do with us moving in one direction or moving towards one of the straits. Okay. So here was our original population, and the solid line is showing what's happening after things. So yes, the solid line does show the phenotypes after the disruption. So directional selection has to do with us moving in one direction. It's moving towards something. Okay. So like horses originally used to be kind of smaller, and after we started using them for kind of agriculture and we used them for travel, we started to see that they were getting larger. Not because of the fact that we use them more, but because those that were larger were more likely to stand up to the strenuous activity, and we saw that they got larger. Okay, not that we made them bigger. We did not do that. Again, don't go marking statements. So Hardy-Weinberg. Hardy-Weinberg is a big topic that y'all all get confused with, and you always ask questions. And actually, I think most of the pre-submitted questions are actually Hardy-Weinberg questions, but that's okay. So with Hardy-Weinberg, this is a kind of a prediction. And in order to follow the prediction and to go with these estimates, there's 5 conditions that need to be met. Okay, and so if you've never seen this video from Paul Anderson, Bozeman Bio, that most of you know him as, he made a video and he talked about it. It's called the 5 Fingers of Evolution. And so in the video, he talks about these 5 different conditions that must be met in order for evolution not to occur. Okay, so we need there to be an extremely large population size. Having an extremely large population size allows for genetic drift to not have an effect. Okay, we have such a large population size that if any random situation occurs, it's not going to adjust or change those allele frequencies. Okay, and for once, I'm actually wearing my wedding band. So the ring finger is our random mating. Okay, so there's no trait that's more favorable or less favorable to mate. It's not like the fish are looking for the more spots to mate with, that the blue-footed boobies having big beautiful blue feet, that those are more favorable. Like there is random mating. Okay, the middle finger has to do with no mutation, so M for mutation, so middle finger. We want there to be no mutations. We're looking for the allele frequency not to change. And so if you add a new mutation to that population, if there's been some change, that allele frequency immediately is going to change. Okay. We don't want there to be any gene flow. So no immigration, no emigration. We don't want any individuals to join our population or to leave our population. Okay. And no natural selection. No trait is more favorable or less favorable than another trait. Okay, so those are our 5 figures of evolution: extremely large population size, random mating, no mutations, no gene flow, and no natural selection. Ow, I hurt my thumb a while ago and that really hurt. Anyways, so you may have heard me say genetic drift a second ago, but like, what is she talking about? So there's 2 types of genetic drift. Both of these are looking at random situations that occurred, and because of those, the allele frequency is changing. Okay, so the first one is founder effect, or founder's effect, as some people say it. This has to do with that there's been a small group of the individuals that have been isolated from the original population. Okay, there was no trait that said, hey, well, this trait is more favorable, so they're going to go over there. It's random which of the individuals from the original population get broken apart and kind of create their own new smaller population. Okay, bottleneck effect. We have this population, there's some natural disaster or a fire, a flood, or something, okay? And that drastically decreases the size of my population, okay? Now, those that survived this fire, flood, whatever, they didn't have a trait that was more favorable or less favorable. So it's random which of those are now going to make up my new population. Okay, so again, genetic drift is going to be a random change in their allele frequencies. Okay, and so once we then reduce that population size, that's when the genetic drift is really going to have even more of an effect, because we could have harmful alleles that now become fixed. When you say that a trait has been fixed or an allele has been fixed, that means that the entire population is homozygous for that allele. Every single individual has that trait. And if every individual in your population has a certain trait and the environment changes and that trait is no longer favorable and is actually unfavorable, we're now going to see a drastic decrease in our population, which could lead to the ending of a population, which is not so good. Okay. So in the equation, okay, again, we're looking at Hardy-Weinberg's equilibrium. Okay. Why are bottleneck and founder's effect not represented lives of the original population? So the reason why they're not like representative of the original population is because there was no, it's not that we selected certain things, it's random. Okay. So if I have 100,000 individuals and 10,000 of them go somewhere, actually 100 of them go somewhere else, there's no like rhyme or reason to those 10. Theoretically, you would think, okay, they would potentially have the same traits, but there could be a chance that they don't. And so since those 100 individuals don't have all those same traits, that's leading to a difference in that little frequency in that new population. So that's kind of really what it comes down to. So back to the equation. So there are 5 different variables in the equation. Your formula sheet doesn't say dominant or recessive on it. I think it says allele 1 and allele 2. So the same thing can be used for if we're looking at things that are co-dominant or incomplete dominant. But I usually use dominant and recessive when I'm talking about this just to make life easier. To help me remember it, the P is a D upside down. So that's kind of how I remember it. Okay. So P, notice I only have one of these. That has to do with the dominant allele. One allele is one letter. Okay. Q is the frequency of my recessive allele. Again, one allele is one letter. Okay. P squared. If I have 2 P's, and we said that P was our dominant allele. So if I have 2 dominant alleles, that would be my homozygous dominant. So P squared, and notice that there's a 2 here, which symbolizes that there is 2 alleles, right? So that would be a genotype or a phenotype that we're looking at, right? So if there is a genotype or a phenotype given to you, that's going to be any of these that have a 2 with them. Okay, that's what I'm trying to point out, the 1s and the 2s. Okay, 2PQ is a frequency of heterozygous. It's a P and a Q. So they have one dominant and one recessive allele, which we know is heterozygous. And then Q squared is going to be the frequency of a homozygous recessive. Again, I have 2 recessive alleles, so Q squared. Okay, and so I always use this little chart whenever I solve them. So P plus Q equals 1. And then P squared plus 2PQ plus Q squared also equals 1. And that's just talking about like the 100% of the population, right? Whenever you solve a problem, you always want to start with Q squared, okay? So this is where we will always start. We find our Q squared, and then what we're going to do is we'll take the square root of Q squared, which gives us our Q, and then we say 1 minus Q, which gives us P, and then we square the P, which gives us P squared, and then we say 2 times our P times our Q, which gives us our 2PQ, okay? Now here's the problem. Based on what you've been given in the question is how we're going to solve this. So I've given you the equilibrium. Okay, equilibrium is an estimation. Okay, now if I know for a fact how many of them are homozygous dominant, how many of them are heterozygous, and how many of them are homozygous recessive, then I'm instead going to count my alleles. So this is not on the formula sheet. This equation we see right here that's called the counting alleles is not on the formula sheet. Okay, it's not on the formula sheet. I'll get your question in just a second. Okay, it's not on the formula sheet. Okay, so this is just the way that I've come up with this formula. You can come up with your own way that will help you out, okay? And I'll get your question in just a second, Alec. And so 2 times my ones that are homozygous dominant, right? So 2 times AA plus the ones that are heterozygous, capital A, little a, okay? Because I'm counting how many of them are that dominant allele. So there's 2 dominant alleles in a homozygous dominant, and then there's 1 dominant allele in my heterozygous. So I'm just going to take 2 times my homozygous dominant plus my heterozygous. That counts out how many of them are dominant. Divide by 2 times my individuals because each individual has 2 alleles, okay? And that will tell me the number of them that are going to, or the frequency of my P. I do the same thing in terms of my Q, so 2 times homozygous recessive plus heterozygous because I'm looking at 2 times my number that have 2 recessive alleles plus the ones that have 1 recessive allele. And again, divided by 2 times the individuals. And then to figure out my P squared, my 2PQ, my Q squared, you just take the number that are each of those genotypes and divide them by my total, okay? So if you are given the number that are homozygous dominant, heterozygous, homozygous recessive, you must use this counting alleles method. If you're given just one of them, you would do the equilibrium. Okay, so someone says, how would you find the frequency of a codominant? Well, if we're looking at codominant, so let's say the snapdragon, right? So with snapdragons, if you have CRCR, that would be red. If you have CWCW, that would be white. And if you have CRCW, that would be your heterozygous, and that would be your pink. Okay. And so I would do that same thing where I would say, okay, well, P squared is going to be my red, 2PQ is my pink, and then Q squared would be my white. And then my CR allele would be my P, and my CW allele would be my Q. So that would just be, you're just determining which one. That's why the formula sheet just says like frequency of allele 1, frequency of allele 2. The second question I was asked is, why do you always start with Q squared? Why not start with P squared? So if you know that it's homozygous dominant, then I guess you probably could start with P squared because that's fine. But if they're telling you that, I don't know, 45% of the population is the dominant trait. Let's say we're looking at, I don't know, black fur versus white fur, and black fur is a complete dominant trait. So we're looking at that 45% are either homozygous dominant or heterozygous. So since I don't know really how many of them are of each of those genotypes, you can't really start with the P squared as your value, because then you try to put 0.45 is your P squared, which isn't actually the true number. So I always find that it's easiest to start with the Q squared, because then you go straight to the homozygous recessive and you don't have those issues. But if you want to start with Q squared, you're welcome. I mean, P squared, you're welcome to. So your answer will be in a decimal if they ask for frequency, and it'll be a percent if they ask you for the percentage. I've seen some questions where they ask you for a percentage and then they say give the answer to the 100th place. And so in that case, they're wanting you to give them an answer from 0 to 1. So you give it to them as a decimal. So they're very clear in how they want you to answer it to them. Okay. So using this will help us to figure out, did our population evolve? Okay. If my allele frequency changes, if my genotypic frequency changes, if any of these numbers change from one population to the next population, evolution occurred. We violated one of these conditions. You got to figure out which one, but one of these conditions was violated if there was some type of change. Okay. So example problem, I just did a quick one, although we're going to do like 3 more problems at the end because they were submitted through the pre-submitted questions. So I'm going to fly through this and then I'll do slower explanation when we get to it in a second. So there are 200 red flowers, 300 pink flowers, 500 white flowers to determine the allele frequency. So in this case, I'm trying to show you why we can't use the equilibrium. And so if we use the counting allele method first, we take the number that are, what am I doing? Okay, so red, we're going to take the number that are red, so 200 divided by our total, which is 1,000, which gives us 0.2. We take our pink, so the number that are pink, 300 divided by 1,000. Again, we're looking at this equation right here, which gives us 0.3. And then with our white, we have 500 white divided by 1,000 gives us 0.5. So then I said, okay, well, we're going to do 2 times my homozygous dominant, which is red. So 2 times 200 plus 300, because that's the number that our heterozygous gives us 700 over the number of individuals. There was 1,000 times 2, so 2,000. So my P would be 0.35. You can still do 1 minus if you wanted to, but I just showed you the other math. So 2 times my homozygous recessive, which was 500, plus 300 being my heterozygous over the 2,000 gives me 0.65. Okay, so that's if you did the counting alleles method, which is the best one to do. Now I'm going to show you if you did the chart and you use the equilibrium, y is going to mess up. Okay, so if you start here with our Q squared, we know it's 0.5. Take the square root of 0.5, you get 0.71, which we already know is off. And then we do 1 minus our 0.71, which gives us 0.29, which is again off. And then we do P squared, which is 0.9, which is again off. And then we do 2PQ, which is again off. Okay, so you cannot use the equilibrium formula if you know the numbers. Okay, how do you figure out what requirement Hardy-Weinberg has broken? Do you have to predict it? There's usually some type of prompt. They'll be talking to you about how, I don't know, they'll probably tell you about these guppies and how the guppies were selecting for ones that had more spots. And they'll go through the whole story. And then, like, you'll say, okay, well, the allele frequency changed. Oh, but they were selecting for the ones with more spots. That was sexual selection, which means we did not have random mating. And so you'll probably be given the information in the prompt to allow you to figure out which one it is. Okay, so phylogeny. Okay, there's a couple different pieces of evidence we could use. You could do biochem or protein. I'm sorry, biochem, which includes DNA or protein. Okay, so you're comparing the number of differences. We also have morphological, so you can look at homologous structures. Okay, so these are the structures that are similar due to common ancestry. So a bat wing and a cat arm, they still have the humerus, they have the radius, they have the ulna. They've got all these same bone structures. Ow, that hurt my thumb. And so because they have those same structures, they are slightly different in functionality, but they are the exact same structures, which shows us morphologically we have these homologous structures. They come from the same common ancestor, and which is why we have those same traits. And then you also can think about ancestral versus derived traits. This question comes up a lot. Y'all ask me. Ancestral trait is a trait that your ancestor had. And so because the ancestor had this trait, all of the descendants have that ancestral trait. A derived trait has to do with something that derived on that pathway. So if we were to think about, I don't know, Dalmatians, okay? Dalmatians on their lineage, and only on their lineage, they derive the having spots, okay? And that, of course, trait. And that came from an activity I did in my class, and none of the other organisms, none of the other dogs we had spots, which is why I went to that. But now that I'm thinking about it, there's probably more dogs out there with spots. So just whatever. We'll just go with that lineage has spots. Okay. So be very careful. So analogous structures are due to convergent evolution. Convergent evolution has to do with that they were in a similar environment. And because they were in a similar environment, a similar trait was more favorable. And because it was more favorable, more likely to survive. And so we saw an increase in that certain frequency of that trait in that population. Okay. So they didn't have a common ancestor. And since they didn't have a common ancestor, although their traits are the same, and although they have the similar structure, they are not related. So just be very careful of analogous structures. So like, I don't know, a butterfly wing and a bat wing. Although they both fly, yay, we fly, they don't actually, they're not the same. Like one of them has bones in it, one of them does not. Like one's an exoskeleton, okay? So big example we use this is the sugar glider versus the flying squirrel. One's a marsupial, one's a placental, so they're really not related to each other in English form, okay? And then biogeography, so looking at where they are in the environment, okay, and what ecosystem they make up, and that kind of helps us to see where they are. Plus, looking at their geological time kind of allows us to see whether they potentially are from the same descendants. So in terms of all of this evidence, what is the best evidence to use? Our best evidence to use, of course, is biochemical evidence. That's our best one to use. It's the most accurate evidence we have. And between DNA and proteins, DNA is more accurate. Reason being, DNA, there could be a change in the intron, which is spliced out that you know from Unit 6. Okay. You could also have a silent mutation. So although the DNA changed, the protein didn't change. And so because there was no physical visible change in the amino acids, my protein is going to show them being the exact same, but my DNA would show them being different. So DNA is definitely more accurate than protein, but biochemical evidence is more accurate than the other ones because morphological sometimes falls into analogous, and then that leads to problems. So they can give you phylogenetic trees, or they can give you cladograms, okay? And so notice that this one is blank. My hypothesis or whatever is that they're going to give you a blank one. This, if they're going to give you one that you fill in, it'll be question number 5 FRQ, okay? And it'll be a blank one like this, and you'll just fill it in, okay? But otherwise, you'll just see like a tree or a cladogram already filled in, and you'll be answering questions about them, okay? They sometimes call them different things, so they'll sometimes call this a cladogram and sometimes call this a phylogenetic tree, but you know what they are, okay? So this one is from, I think, the 2018 exam, and they ask them questions about the bears and how they're related to each other, but I think I'm going to go down the cladogram first. So in terms of my cladogram, if I had to fill this in, I'm going to look and say, okay, which ones are most closely related? So the black bear and the brown bear, I see have one difference in their amino acid structure. So that tells me that the black bear and the brown bear should be close together. So notice that the V shape you have here is a very small V. So that means these two, that's where we're going to see the black and the brown, okay? And then if you look here, the panda has 33 from black, 34 from brown, and 40 from polar. So that's showing us that there's a large difference between the panda and the black, brown, and polar bear, okay? And so we would put panda bear here, the polar bear there, the black bear there, and the brown bear there, okay? So someone says, could you explain what each line means? So this is just showing the lineage. So this is where the common ancestor is of that panda, and it diverged off to become what we now know as the panda. This is where we would see the polar bear's descendant, okay, kind of branched off that made our polar bear, and so on and so forth. Someone says, can you have more than one answer? Yeah, I graded a free response question many years ago, probably my first, second year grading, in which there were 4 different answers for the phylogenetic tree. So we had to know all 4 answers. It was so much fun. I think I actually have that one coming up soon. So if there is a branch point, so right here we see the black and the brown, there's a branch point, and there was no evidence to tell me what should it be at the end and what should be, in the middle. So the black and the brown could have been substituted, and that could have been brown, and that could be black. So you can rotate those two. So One of the questions that was asked was which ones are closely related. So the European brown bear is closely related to the polar bear because it shares a branch point, as well as the Western North American brown bear and the Asian brown bear are closely related because they share that recent common ancestor. You can rotate this. You could have put Asian here and Western North American here, as well as you could have put polar bear there and European brown bear there. But you couldn't rotate around those two if you didn't want to, because notice that they're common ancestors at different points in the lineage, okay? If you want to take the entire thing, I guess you can switch that, but again, you're not really changing anything, okay? And then in terms of what could rotate here, the black and the brown could rotate, as I said before. Okay. So the question they asked was how many years ago was the common ancestor of the brown bears? So you notice that we have brown bears here, brown bears there, and brown bears here. So you go back to where was the most recent ancestor, and that's that branch point we have there. Drop it down, and so it's like 3.3 hundreds of thousands of years ago. Notice your axis goes from 50 to 1, so don't misread this as 4 point something, which I believe a lot of students may have made that error. So make sure you're watching your axes whenever you're giving some type of data from a chart. So, speciation, okay? So this is where we create new species, okay? So biological species concept states that two individuals are of the same species if they can interbreed and produce fertile, viable offspring, okay? So that's really what this comes down to. Interbreeding looks at our prezygotic, and then the fertile, viable offspring looks at our postzygotic, okay? So with our prezygotic, this is before the zygote, pre making you think of before, zygotic makes us think of zygote, so when sperm and egg come together. So our behavioral, this is ones that have two different behavioral rituals. So as I mentioned before, the blue-footed booby, it does a beautiful little dance. It shows off his blue feet. You know, you want to mate with me? I have blue feet. So that would be a behavioral difference. If a red-footed booby walks up to the blue-footed booby and tries to do a dance, that blue-footed booby is like, no, 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 thank you very much. We also have temporal. This has to do with that they mate at different times. So different times of the day, different times of the month, different times of the year. If you follow Marshmallow Madness, you saw the Eastern skunk. So there's an Eastern skunk and a Western skunk. They actually mate at different times of the year. I think one's in the summer and one's in the winter. But because they mate at different times, they don't mate with each other. We also have geographic. So there's some type of geographical barrier that stops them from mating with each other. And so because there's that barrier, they don't come in contact with each other, so they don't mate. We also have habitat ecological. So they're in different ecological environments. There's no actual barrier. So if you were talking about like there's a snake in a tree and snake in the water, and so the tree snake and the water snake aren't really going to mate with each other because they're in different habitats. There's no actual barrier. Like the snake in the water can climb out and go up the tree, and the tree one can fall in the water. But they're probably not going to come in contact with each other because they don't really are in the same spot. This happened with, I think there was like a hawthorn maggot fly and an apple maggot fly. And because they selected different trees to feed in, they actually didn't mate with each other. Mechanical, this has to do with that they're anatomically incompatible with each other. There's some snails. Gametic, this has to do with the two gametes are unable to fuse. So this is usually external fertilization. So like sea urchins that lay their eggs along the bottom of the water, the sperm isn't going to fuse with the egg because they're two different gametes or two different species. They're not going to have the right glycoproteins on the outside of their membrane, and so they're not going to be able to fuse to fertilize that egg. So postzygotic, post is talking about after the zygote is formed, so postzygotic, okay? So after we create that zygote, so we can have reduced hybrid viability. So the hybrid is not viable. As you said here, like biological species concept, they interbred, check, okay? But they didn't produce viable offspring. So since those offspring are not viable, those two species are not actually the same species. Okay, reduce hybrid fertility. So the hybrid is not fertile. Okay, again, we need them to be fertile offspring. So this is the horse and the donkey example. If the horse and the donkey mate together, they make a mule. A mule. I like to call them horkies, but that's not really what they're called. But so we have a mule, and the mule has an odd number of chromosomes, and since it has an odd number of chromosomes, it can't pair up its homologous chromosomes for meiosis, so it is unable to make those sperm and eggs to be able to reproduce, so they are sterile. And then hybrid breakdown. The first generation, hybrid's okay. But as that hybrid continues to try to mate, we're going to see a decrease in its viability. I know the camera's in the way. I post the PowerPoint later. I'm so sorry. But those three were reduced hybrid viability. The hybrid is not healthy or viable. The second one was reduced hybrid fertility. The hybrid is not fertile. And I think you can see the third one of breakdown. And I'm sorry the camera blocks it. I found that it's just, if I keep popping back and forth, I sooner or later cover myself up again. So there's two different types of speciation. We have sympatric speciation. The S in sympatric makes us think of same. So we're in the same place, okay? So two species of them are going to continue to inhabit that same geographical area. The mule has 63. That wouldn't, as I said, the mule is going to be the reduced hybrid fertility, right? They're not fertile. They have an odd number of chromosomes. They can't make gametes, okay? So sympatric, they're in the same spot, and so we have them in the same place. And so since they're in the same place, there's going to have to be more of these pre-zygotic barriers to inhibit them from coming in contact from each other and mating. So habitat isolation, behavioral isolation, sexual selection, and then polyploidy. Polyploidy has to do with that we have more than two sets of chromosomes. So like bananas are triploid, and like wheat I think is a tetraploid. Like so there's a lot of polyploid species out there. So here you can see the apple maggot and then the hawthorn maggot or blueberry maggot. Anyways, they don't mate with each other because they're on different plants. Allopatric has to do with that there is a geographical barrier. So they're not going to mate with each other because of the fact that there is a geographical difference. There's something that blocks them from each other. And so these two chickmunks, squirrels, whatever they are, aren't going to mate with each other because now the Colorado River is between them. Okay. Whew. Hey, I did pretty good. 35 minutes to get through all the content. So I asked students to submit questions in advance, and so these were the questions that were submitted. So someone says, can you dumb down Hardy-Weinberg math problems? I will do my best to dumb them down, and so we're going to dumb it down through the usage of a question from College Board. So this is from the 2013 exam. It's number 53. I actually did it on my math minutes the past 2 days, but that's okay. So there's a syndrome. It is a recessive genetic disorder. So that tells us that if they have that syndrome, that means they're homozygous recessive. And it gives us information about that disorder that we don't care about. In general, the population, this syndrome occurs approximately in 1 in 150,000 live births. So the general population, 1 out of 150,000, is going to be the Q squared, okay? And then in an isolated population, instead we have a 1 out of 500. So again, we're looking, this shows us like technically showing us founder effect because I had a small population that got isolated, and now I can see that those allele frequencies are different, but that's okay. So we're now going to assume that they're in equilibrium with respect to the syndrome, and then we need to talk about the differences in their allele frequencies, okay? So they gave us a lot of answer choices. I'm not ready to use those answer choices just because of the fact that we haven't done any math yet, okay? So here are some quick tips when we're doing Hardy-Weinberg. So number one, look at the information. Do you have all the numbers of the genotype? Do you have the A little a, and then little a, little a? Okay, so do you have them all? And if I look here, I see I have 1 out of 150,000. That only tells me like my Q square. That's only telling me one of them. That's only giving me a phenotype, right? So the second one is, or do you just have the information about a phenotype or an allele? I just have information about a phenotype or an allele. So I'm going to use the Hardy-Weinberg equilibrium equation, okay? And I was given a phenotype. So I either have the dominant phenotype, which is a p squared plus my 2pq, or I have the recessive phenotype with my q squared. So since I look back at the question, I see, oh, this is a recessive trait, and I have the value of 1 out of 150,000. That means that I'm looking at my q squared, and I'm going to use the Hardy-Weinberg equilibrium. And again, I want to start with q squared, and so that's really where we already are. So we're going to start with that, okay? So I rewrote my numbers there just to make life easier, and then I have my beautiful little chart. Yes, you know I love my chart. So I made it so there was one column for general and one column for isolated. Okay, so we start with q squared. 1 out of 150,000, there we go, is going to be my q squared. I typed that into my calculator, and my calculator tells me that it is 0.000006. Whew, that's a lot of zeros. So because of the fact that you get your TI calculator or you're using your Desmos, okay, because within the Blue Book system, you're going to have Desmos, you're then going to take the square root of that number. Okay, so take the square root of the number you just had, you know, control, answer, all that fun stuff. And that's going to tell us that our Q is 0.00258. Now, do I need to solve for P squared, and 2PQ? Well, no, because they just want to know what the allele frequency is, right? If I look here, what is the frequency of the allele that causes the syndrome? I only need to know what Q is. So I'm actually not going to solve for P squared, or 2PQ, okay? So now let's look at the isolated population. So in the isolated population, I had 1 out of 500. I go ahead and sub that into my calculator, and I get 0.002. And then I get my Q value, take the square root of Q squared, and that's going to give me 0.0447. Okay, so all I did was take my Q value, my Q squared, which was given to me in the prompt, and I took the square root of it to get my Q values. That's all I've done. Okay. Now I just combine that chart so I can fit it into one spot. And I'm looking at my answer choices. The frequency of the allele is 0.002 in the isolated population. No, it's not. The frequency of the allele is 0.0447 isolated population. Hey, yeah, it is. Cool. And 0.002 in the general population. Hey, yeah, it is. Cool. So B is good. Let's go on to C. The frequency of the allele is 0.00. No, it's not. And then the same thing here is 0.0447. Yes, we know that is true. So now I have to finish looking at the question, right? So showing the rate of genetic mutation is highest among individuals in the isolated population. Well, it said it was in Hardy-Weinberg. And based on the 5 conditions of Hardy-Weinberg, I know I need there to be an extremely large population size, random mating, and no mutations. So B is not logical because I can't have any mutations since I'm in Hardy-Weinberg. So I know that B is out. And then D says, in which suggests the genetic drift occurred in the isolated population. So there was a smaller population that got isolated from a larger population, and that has allowed it to get a different allele frequency. So D is our answer. Okay. Someone says, okay, so let me look at the question that we have in here. Off-topic question. If an FRQ doesn't explicitly state which type of graph to use, does it matter which one you use? Yes, it does matter. You're going to have to use the data you've been given to determine what kind of graph to make. So if we're trying to figure out what kind of graph to make, if you have categorical data, big example would be there was a muscle question from 2015-ish. They told you had 8 different sites that you collected samples at. Those sites are categorical. They don't actually like connect to each other. Like they're just sites in the water. That's categorical. Versus if you have linear data, that's going to be a line graph. So categorical is bar graph. Linear data is going to be line. So if you have temperature taken every 5 degrees, you have time every 5 minutes, you have something that is like individual times. Now, you can't just assume time because there was the caffeine question from 2016, 2017, in which you had the like 10 minutes and 24 hours. Those numbers are so far apart from each other, you cannot put them on a linear scale. So that was a bar graph. And then if you have 2 things you're comparing, like number of alleles to number of genes, or I don't know, number of DNA mutations and number of alleles, or something of that nature, right? And then you have individual like that are following that, and you're looking at the trend between genes and number of alleles, or whatever numbers I said. That's going to be a scatter plot. Okay, so those are like the 3 to make sure we know. Box and whisker is in the CED as a potential graph. So you graph the middle line is going to be your mean. And then the first top of the box is the 25% of the data, and the back part of the box is the other 25% of the data. And then you'll have a line that comes out, that's another 25%, and then a line that comes out, and that's the other 25%. If you look at the 2022 number 6, that question shows a box and whisker. If I remember correctly, it's either 2022 or 2023. One of those 2 is a box and whisker that you could use to practice. Anyways, and so those are how you figure out the graph. You're welcome. Okay. No, I don't have any extra worksheets on Hardy-Weinberg. Hardy-Weinberg is one of those ones that there's tons of practice online. Google Hardy-Weinberg practice and key, and you're going to find tons of practice worksheets. Okay. So when constructing your phylogenetic tree, how do you determine where to place the traits that show the differences between the species? Should I place it on the branch that disconnects them, or should I place them on the branch for that specific species? So you're going to place it on the branch going to that thing. So the difference between the lamprey and the sea bass is the sea bass has jaws. So then, of course, the antelope, the bald eagle, and the alligator all have lungs, but the sea bass and the lamprey does not. So always think about these as like you're driving down a road. And as you drive down the road, you pick up these different traits. We drove down the road and we turned. And so none of the traits are in the lamprey. And of course, that would be considered our outgroup. We drive down the road, we pick up jaws, we pick up lungs, and ooh, look, we picked up fur. Well, we're an antelope. We picked up jaws, lungs, gizzards. We're an alligator. We picked up jaws, lungs, gizzards, and ooh, feathers. Now we're bald eagle. Like, so you're picking up those traits as you go. You can also drop off traits. So like if everything has a certain trait, you could then see loss of a trait. Which year was it? I can't remember off the top of my head. I think it's like 2016, 2017. There was, or maybe 2018. There was a question about like it was evolutionary question and the immune system question, and it was talking about like having lactose, having protein A, protein B, and casein. And so with those, you were talking about where they picked up those traits. So you would just either mark them on there as gained or lost. So this question is from 2013, number 5, and I just want to show how you would mark those on there. So they said using the template. This is how you'll see number 5C is going to be giving you some type of like diagram, and you're going to have to modify that diagram. They said represent the points at which characteristic 1 listed in table 1 evolved by marking X on the line of the cladogram in the correct locations. So if we look here at number 1, we can see extra tooth material. We can see the bovidine or whatever it is, and the anthrocappa, whatever it is going to get the X. Okay, so bovidine is here and the A word is there. So if I put an X there and I put an X there, so you're just putting the X on that lineage going to that that group or that individual. So I hope that answers that person's question. Okay, so then it said also looking for an explanation on dealing with constructing phylogenetic trees with molecular data. So I figured the best way to talk about how you can use molecular data is to do an example of one of constructing a tree, which I made it a cladogram instead because that's what I had access to. So we have the amino acid sequence of chromosome C, 5 different species of vertebrates. They gave them in a table and then used the table to create a phylogenetic tree on the template. Notice this is an older question, so this is 2015, and so it's slightly different in a way. So this is the chart they gave them or the cladogram they gave them, and then this is the chart. Okay, so they're asking them to fill in on here based on what they know. Okay, so we can see that E. africanus and E. ferus is going to have 1 amino acid difference, so we can put those together. Again, they're going to be close together on a branch because of the fact that they only share 1 difference. And if I look here, I see there are 3 differences between A. forestry and G. gallus. P.S. G. gallus is a chicken and A. forestry is a penguin. Shh, it's okay, no worries. So we put those together because they have 3 amino acid differences. And then the one that doesn't share any of them, as you notice here, was it D. It has 21, 18, 17, and 20. So we know that one's going to be the last one. Okay. Now this question actually, because of the fact that there was 2 different branch points here, there's 4 different answers. It could be D, G, A, E, E. It could be D, A, G, E, E. Or you could switch these back too. You could also rotate it, and it could be E. Yeah, it could be E-E-A-G, E-E-G-A. Like you could have all types of rotation. There's lots of things you can do with that. Someone says, with the exam being hybrid, how can we mark cladogram or other data sheet will be provided in the FRQ booklet? Okay, so you in your hybrid exam, so in case you're not aware, your multiple choice is going to be on the computer and you're going to answer on the computer similar to how you did your PSAT and your SAT this year. Okay, so you will get, I think, up to 5 sheets of scratch paper, so you can mark on your scratch paper. And then in terms of your free response, you're going to have the questions on the computer, and then you will have a physical book. So on question 2, you'll have the blank graph paper in your book. And then on question 5, you'll have the blank whatever you're annotating. So you know, last year or 2 years ago, it was that cladogram that they had to build on. Last year, I think it was a phylogenetic tree that they had to build on. So they're giving you something to build off of. Okay. Other question, off topic. If you are someone who likes to pick and choose whichever FRQs to answer, how much space would you leave between each page? Okay, so I'm glad you asked this question. I'm going to go ahead and make this statement, and I want you to listen very closely. Very, very closely. Everyone listen very closely. Okay. Your booklet is pre-labeled. When you flip the first page, it's going to say answer for question, or response for question 1. And then a couple pages later, it'll say response for question 2. And response for question 3. Okay. There are specific pages designed for specific questions. Okay. Now you as a student, you get rolling with 1, and then you flip the page, you get rolling with 2, and you accidentally write question 2 in the space for question 1. I need you to listen very carefully. Take a breath. It's okay. Go to question 2 where you were supposed to write it and say, see question 1 for FRQ number 2. Do not, I repeat, do not rewrite your FRQ in question 2 spot and put a big X over it in question 1. Okay. Your readers will have access to your entire book. Okay. So in answer to your question, how much space do you leave? You will just go to whichever question is that you're answering and don't worry about leaving space for it. If you're liking to answer C and then you want to answer A, then you want to answer B, just write it out as C, then A, then B. You do not have to answer them A, B, C, D order. You may answer them in whichever order you want on that page. But just label it for us. That'd be really helpful. So if you just mark like C, I, and then you write out what your answer is, C, or A, sub I, you write out your answer. Like just clearly label it. It just makes it easier for the readers, but you don't have to label them, and you don't have to answer them in order. But it does make it easier for us if you at least label them. Okay. So someone says, I feel confused about Hardy-Weinberg. I don't know how to use the first one versus when to use the second one. So can we do a bit of practice? Okay, so here let's do some practice. This one also is from the 2013 exam. It's number 121. It's 121 because the numeric response was a different numbering. There's no numeric responses on this current exam. So some people have the ability to taste a bitter chemical called PTC. The ability to taste it is due to presence of at least one dominant allele. Okay, so being a taster is dominant. The incidence of non-tasters in North America is approximately 45%. Assume populations are Hardy-Weinberg. What percent is homozygous dominant for the ability to taste PTC? And then provide your answer to as a number between 0 and 1 to the nearest hundredths place. Okay. So what have I been given? I've been given the number of non-tasters, a percent of non-tasters. I do not have the number of homozygous dominant. I do not have the number of heterozygous. All I have is homozygous recessive. So I'm going to use my Hardy-Weinberg equation. Okay, so I use my little chart, and since the 45% is my Q squared, I fill in Q squared. And then we did this before. To get from Q squared to Q, we're going to, everybody now, do the square root. So the square root of 0.45 will give us 0.67. Now if I know Q, now I can find P. How do we find that? 1 minus my Q gives me my P. So 1 minus 0.67 gives me 0.33. Now that I know P, I can square that value. So 0.33 squared is going to give me 0.1089. And just for kicks and giggles, we're going to finish our chart. So 2 times my P times my Q is going to give me my heterozygous, which is 0.442. Now the question says that I want to know the homozygous dominant, which is this P squared right here. And I want to give my answer between 0 and 1. Ooh, I'm going to use a decimal. To the nearest hundredths place. Hundredths place is 2 decimals. So that would be 0.11. So that's kind of looking at that question. We can see that. My son helps me come up with those questions, so I know it's a little weird, but that's what my child came up with. So this is what we're going to do. Population of herons has incomplete dominance shown in 3 different phenotypes. It could have a yellow head, which is YY. It could have a white head, which is Y little y. It could have a gray head, which is 2 little y's. So during spring break, you sat on a dock, and you observed 7 that have yellow hats, 3 with white heads, and 1 with a gray head. Oh, yeah, I'm good. Okay. Determine the frequency of the Y allele in the population to the hundredths place. Okay, so I know how many of them is my homozygous dominant. That's 7. I know how many of them are heterozygous. That's 3. I know how many of them are homozygous recessive. That's 1. So I'm going to do the counting alleles method. Okay. So since all I have to do is figure out the Y allele, I'm just going to do the equation for P. Okay. So I'm going to say, okay, well, 2 times my number that are homozygous dominant, which is 7, plus my number that are heterozygous, which is 3. And I'm going to divide by the total, which is 7 plus 3 plus 1, which is 11, times 2, which is 22. So 7 times 2 is 14, plus 3 divided by 22. That gives me 17 over 22, which is going to give me 0.77, which is actually the answer that I was looking for in this question. Is the percent always Q squared? So somebody just asked, is the percent always Q squared? No, they could say like the incidence of tasters is 45%. And in that case, you would then say 1 minus 45%, and you would find that Q squared is actually 0.55. So you have to use the information from the prompt in order to figure that out. Okay, so that gets us through that. So someone says, why does random mating not change the allele frequencies? So if your class did the lab in which you used the beads in a cup, that cup had the same allele frequency in it. And so random mating is pulling from that same gene pool. And so if in your gene pool you have 50% black and 50% red, then when you shuffle that population up for the next generation, you still have 50% black and 50% red. You're going to maintain that that allele frequency because of the fact that it's still the same gene pool. And so it's just randomly pulling 2 alleles from that gene pool is really what it comes down to. These pictures come from that 5 Figures of Evolution from Paul Anderson, as I said, both in bio. much better explanation for it than I just gave, but it really has to do with it. We're pulling from that same gene pool. And then someone says, what is fitness and how does it relate to natural selection? So fitness has to do with the contribution that you're making to that next generation. So the mule, as we mentioned before, that is sterile. It has a fitness of 0. It cannot have offspring, so it's going to have a fitness of 0. It doesn't contribute to the next generation. It doesn't contribute to that gene pool. Okay. So this one right here, the green bug, has a lower fitness because it only has 2 children that end up having, another 2 children. So there's only 4 grandchildren versus this one has 4 offspring each time. And so there's a higher fitness. We have more of those brown alleles in that population because each time they're more likely to survive and more likely to leave more offspring. So, and then how can you explain how Darwin explained natural selection? I truthfully don't know how he came up with it, so I had to Google it. So I found an article that talked about that he took a bunch of notes while he was on the SS Beagle. I'm sorry, HMS Beagle. And so on it, he kind of was making observations about the finches and about their beak sizes. And so the ones that had different beaks, they lived on different islands and they had different environments. And so like due to that, he was able to then make a connection between how the environment influenced those that were more favorable. He also saw that the ones that had that were best adapted to get more food, and so they had the advantage. And so he saw that there were greater numbers of them in that population. And so eventually they formed their own distinct species of that one, and that's when he really came up with the survival of the fittest, where fitness has to do with that contribution to the next generation and the more favorable traits. So that brings us to the end of the pre-submitted questions. So if you have any other questions about Unit 7, please let me know. As a reminder, I just want to, kill some time. So this is my website, appenguins.weebly.com. There is a ton of resources on here. I mean, like literally a ton. So if you're looking for the most helpful tools, right? Because we're in the final crunch, 3 weeks of the exam. Last year's students, when they answered the survey to tell me what was helpful for them, they said the unit review videos were helpful, the TikTok videos, and then the quizzes games. So here is where all of those are. You can also access tons of resources. My review guide is here, the 296-page review guide. FRQ Fridays, this is where every single FRQ from 2013 to current is, where I've basically walked through every single one of them and explained them all. There's the PowerPoints for all of my reviews. So if you've missed the Insta-Review on my Instagram, this is where all the PowerPoints for it are housed. So you can actually still access them all. These are resources from other students. These are the math videos. I've gone and made 1-pagers for every single unit. And then if you want to be nosy and see how last year's kids did, you can press on the past Penguin results and you can see what resources they used. And also you could see like what was most helpful to them in that exam or in that review time. And you can actually, see tons of like literally just go through. There's tons of resources on here. I've been doing this whole thing since 2019 in terms of the website. Like it's tons. Anyways, okay. Can I go through the format of the exam? Yeah, of course I'll go through the format of the exam. Okay. So the exam has 2 portions. You have 90 minutes and you're going to take a multiple choice portion. There are 60 questions on the exam in terms of multiple choice. So there's 60 multiple choice questions and you have 90 minutes. So that's about 1.5 minutes per question. Not every question is going to take you 1.5 minutes. Not everyone is going to take you 1.5 minutes to solve. So I would probably get every 10 questions, have you done 15 questions. No, I said the backwards. Every 10 questions, have you gone 15 minutes? Okay. So make sure that you've only elapsed 15 minutes because if you've elapsed more than 15 minutes, you're moving too slow. If you have less than that, then you're good and making good pace. Okay. Then you'll have a quick break. I believe it's 10-minute break in between the 2 sections. And then you'll have your fair response. Fair response, you have 2 long fair responses that are going to be 8 to 10 points each, but together will equate to 18. So you could have an 8 and a 10 and an 8, or 2 9s. You can usually tell the number of points based on the bolding. And then most recently, they started kind of doing the A sub I to ensure that you see all of your stuff. So you can kind of use that as a way to track how many points there are. And then your short FRQs are all 4 points each, and that'll be questions 3 through 6. In terms of pacing, I would spend about 20 minutes on your long FRQs and about 8 minutes on your short FRQs. So as soon as you get to your, you press the button and you open up the exam, first 8 minutes, I would read through your questions and find out which ones you want to answer. I would pick 1 long and I would pick 2 shorts. and then I'd have one long and then the other two shorts. And that way you're ensuring you're getting those long FRQs answered. And then I would renumber them and do them in the order that I know them. Then I would do my long one, as I said, 20 minutes, and then my short ones, 8 minutes each, and then go back to the long. And that will give you 10 minutes at the end that you can go back to any of your questions and add anything that you feel like you need to add. If you ran out of time on a question, you could go back and finish answering that question. And of course, this is just a rough estimate. If you want to spend 25 minutes on one of your FRQs, go for it. Okay, I'm not telling you not to do that. I'm just telling you that this is a way to kind of pace it out to try to at least get something on every question. Okay, so what kind of structure should you know? Homologous structures, analogous structures, vestigial structures. So yeah. All right. You have the Pearson, Princeton, and Barron's prep work. Which one do I think you should use for practice tests? They're all equivalent. I really don't feel like any of them have a better practice test than the others. I don't know if Pearson's does this, but I know for a fact that Barron's prep book has that the exam is broken down. And so after you do the practice exam, you can see where your strengths and your weaknesses are. So I almost think the Barron's is better because you know, oh, after I finish this exam and I analyze it, I know I struggle with unit 5 or I struggle with unit 7 or whatever unit it is that you struggle with. So any other questions? If not, I'm going to end it because I am at my 1 hour that I said I was going to make. So, going once, going twice. Okay. Remember, Eighth Bio Pain was just success. I hope that y'all do amazing on May 5th, or Cinco de Mayo as we're all calling it.